Okay. So, good evening. Welcome to the week 9 live interaction session for the immunology course. I'm Ankita and I'm your TA for this course. And as per our usual discussions, today we will be first covering the assignment questions for the last week, for week 8, followed by which we will go over the topics that were discussed as part of this week in more details. And I have different schematics and different additional material for you to understand the same material a little better. And finally, we will conclude with some of the sample questions as were given in the previous year so that it helps you in your preparation. And we will address any queries or any doubts that come up during this discussion at any point of time. I can be interrupted. Please do interrupt me if you have any queries, and I'll try to address them. If it is already in my slide material, we'll take it up today. If it needs additional help, of course, I'll try and get better materials to help you understand that concept. So again, before I start, most of the slides material that you'll find has been taken from Kubi. That is the main book I refer to. You can refer to any of the suggested books by the professors, especially Janeway, and of course to the slide material that is already there in the YouTube lectures, which you have found uploaded. So with this discussion, let us first get into the previous week's assignment. So we had discussed the process of entire B cell maturation and the way in which T cell effector mechanisms are brought into place in the last week's discussion already. So I will go back and forth over the slides because I don't have the schematics here to in case some reference is required. So in the first question, we were asked that multipotent progenitor cells via their FLT3 receptor tyrosine kinase bind to dash cells. Which of the cells do they bind to? Stromal cells, mast cells, dendritic cells, or macrophages. So give me a second. Yeah. So it was there in this slide that if you remember that we had talked about the way in which B cell maturation is taking place. But yes, we got into the details of it much later, how the development occurs. But if you remember the initial stages, then the process of development from a bone marrow stromal cell, from an undifferentiated or a cell which is yet not destined its fate, from that condition to its development into a pre-B cell, a pro-B cell, pre-B cell, and finally into an immature B cell, which is whose antigen specificity has been defined, but which has not yet met its antigen. That is a naive but immunocompetent B cell. That entire process was discussed. So as part of that, we had seen that this initial interaction that occurs between the B cell, the developing B cell, and the bone marrow stromal cell is in this following manner, and that involves interactions between the receptor tyrosine kinases, and FLT3 is one of them. It is not here in this schematic, but if you remember the one which was given as part of the weekly lectures, then one of the other very key interactions involved the FLT3 interaction for their development. And the cells with which they actually facilitate that interaction is with the bone marrow stromal cell. So that is the correct answer to this question. Coming back again to that slide. So which chemokine, the second question was about which chemokine helps to retain stem cells and lymphoid progenitor cells at appropriate stromal cells in the bone marrow. So we had discussed some very brief introduction to chemokines that already been done. We will be discussing cytokines in more details furthermore, and there we'll actually come across the different types of interactions that occur within the immune system, which helps in which helps in additive or subtractive or synergistic or pleiotropic effects, etc. We'll learn about that. But briefly, what we had, oh, so sorry. Briefly, what we had discussed was the role of these interactions in facilitating, for example, let me give you an example. Yeah, we talked about chemokines and cytokines and how they help in cell and intercellular and intracellular interactions. So we had talked about autocrine interactions, where one particular cell is synthesizing a chemokine. Yeah, one particular cell is synthesizing a chemokine or cytokine, which is acting on its own cell, versus when it is synthesizing a cytokine and releasing it into the external environment, and then that cytokine goes and acts on some other neighboring cell, then that is a paracrine interaction. So all these different types of interactions will come to very soon. One of the chemokines which we had gotten familiar with was CXCL12, and that helped in the retention of stem cells and lymphoid progenitor cells close to the bone marrow. So CXCL12 was the correct answer to this question. We had discussed it already. And just from the diagram that I showed you just before this, we had seen how pro-B cells mature into pre-B cells. And that interaction involves which of the following receptor ligand interactions. So that has A, VLA4 and VCAM1, B, c-KIT and SCF, stem cell factor. C is for A and B, both options, or FLT3 and FLT3 ligand. So I'll just go back to it very quickly. So these are the interactions that are being mentioned. That is VLA4 and VCAM1, as you can see here, and c-KIT and SCF, as you can see here as well. Both of them are necessary for the conversion of a pro-B cell into a pre-B cell eventually. So with this, the correct answer, therefore, in this case, would be A and B. Both options are correct here. Coming to the next question, the heavy chain rearrangement occurs in the A, hematopoietic stem cell, B, multipotent progenitor cell, C, pro-B cell, or D, pre-B cell. Again, if anybody has any doubts, please do interrupt me at any point of time, and I'll try to address that question, just if it is confusing. So yeah, so the question asked here is about heavy chain rearrangement. So if you have to read the question a little properly, whenever we are talking about gene rearrangement or heavy chain rearrangement, it means that it is occurring within the genes which are coding for the antibody or for the B cell receptor. And obviously the B cell receptor fate comes up a little later. I'll go back again. I think the slides and the schematics are always more helpful in this regard. So yeah, this is sorry, this is for T cells. Just a second. If we can find, just need to go back to that slide. But yeah, I'll just verbally try to explain that if you remember the way in which B cell receptor fate comes about, I'm not able to find that slide right now. But anyway, that's fine. We can address the question either way. That whenever the gene rearrangement becomes a question, then heavy chain gene rearrangement will occur in a cell which is already fated to be a B cell and is in the process of development. So obviously, by default, the correct answer will obviously not be option 1 or option 2, because hematopoietic stem cell or the multipotent progenitor stem cell are more primitive forms or the precursor forms of the developing cells. It will be between pro-B and pre-B cell. Now, to get more to accurately answer this question, which of the two would be correct, you have to know the stages at which gene segment rearrangement occurs. And among these, the pro-B cell is the stage where heavy chain gene rearrangement starts taking place. So the correct answer is pro-B cell. So it already begins when B cell fate determination is occurring. So this would be the correct answer. You can find the reference to this from the slides, or you can find it from any textbook that you are referring to at this point of time. In the next question, we were asked about matching the B cell stage with the proper description. So this is actually going to help you address the previous question. So I'll directly get to the answer because it's easier to understand that. So the different stages have been divided as early pro-B cell, small pre-B cell, immature B cell, late pro-B cell, or the large pre-B cell. So while we have not gotten into these intricate details between the within the lectures or even within the discussion session, you can find this online for your reference. If there's any confusion, I can try including a schematic and we'll discuss that in this week's discussion session for week 10. But for now, just for an information basis in a factual manner, what you can keep in mind is that the staging is such that option A, as you can see, is matching with option 2. So early pro-B cell has the heavy chain DJ rearrangement. So if you remember the sequence of events in which rearrangement occurs, it starts with the diversity and joining segments, them undergoing a gene segment rearrangement, then comes in the variable region. So that is the correct option. A2 is the first correct option. Coming to option B, for the small pre-B cell, you have the correct option as option 4, where VJ rearrangement is occurring within the light chain. You need to keep in mind the fact that each of these options has heavy and light chain both, so please don't confuse the two. When we talk about VDJ rearrangement for the heavy chain, that is occurring in a different stage, that is occurring here in the late pro-B cell. So if we go sequentially, first comes the early pro-B cell, where you can see DJ rearrangement is occurring in the heavy chain, following which VDJ rearrangement in the heavy chain is coming in the subsequent step, which is for the late pro cell. So that is D1. So if you get these two options correct, obviously you will eliminate all the other three. Now comes the finer details of the remaining options. So we have option C, which is the immature B cell that has surface IgM. Now, what you need to keep in mind is that the immature B cell is it's towards the fag end of the developmental stages, which means that it has undergone all the previous rearrangements. It has now come to a state where it is capable of interacting with its antigen and then eliciting the downstream signaling pathways, which will help in removing that pathogen from our circulation. So therefore, it is at a stage where you can understand that its fate has been determined and what it is expressing on its surface is a surface IgM. So for option B, the correct option is 5. And yeah, so with that, we option C, the correct option is 5. So option D obviously has only one option left here, which is for the late pro-B cell, and that is that is what we have already discussed. And finally, for the option E, large pre-B cell, the correct option remains as the expressing pre-B cell receptor. So it is a precursor stage to the final immature B cell, and therefore that is the correct option. So with this, you can address this question. The following questions were about B cell activation. It occurs in the A spleen, B lymph nodes, C thymus, or D bone marrow. So again, keep the terminologies very clear in your mind. When you talk about activation, that takes place within the lymph nodes. When we talk about development, when we talk about maturation, they're all in different locations. So what the question is exactly asking, you have to address specifically that. So option B in this case is the correct option. The next one was about protein antigens that can induce B cell activation by dash. So when you talk about the type of antigens, they can the way in which they can elicit an immune response, that will be different. When we talk specifically about protein antigens, they do so by a thymus dependent process. So we had discussed this in the last session as well about T dependent antigens or T cell independent or thymus dependent or thymus independent forms of activation. So option B in this case is the correct option, which is why it is a thymus dependent process. The next one was about the example of a type 2 thymus independent antigen is or are which of the following. So again, this is a very it's a factual question. If you know the answer, you'll be able to address it. So it is about A, lipopolysaccharide or the LPS, bacterial flagellin, bacterial cell wall polysaccharides, both A and B or both B and C. So if you remember, we had a table which was addressing this. So with respect to that, it has both options B and option C as the correct option. So flagellin as well as cell wall polysaccharides of bacteria can induce the thymus independent form of an immune response, which is therefore they are TI2 types or thymus independent type 2 of antigens. So therefore, correct option is E. And finally, for the last two questions, we were asked about given are the steps for maturation of progenitor B cells into precursor B cells, the correct sequence of events are which of the following? So we have just seen the interaction, like we saw that schematic which shows the types of interactions which occur. So among them, the first interaction which takes place is between VCAM1 on the stromal cell and VLA4 on the pro-B cells. Option 3 becomes the first step. Following which you have c-Kit and SCF interaction. If you remember that diagram once again, therefore option 1 becomes the next sequence of events. Third is the expression of IL-7 receptors on the pro-B cell. Again, you can refer to that schematic for your clarity. Yeah, following which we have binding of IL-7, which has been released from the stromal cell onto the IL-7 receptor. So after expression has occurred, there is interaction between IL-7, IL-7R or IL-7 receptor. And then obviously the final step will be maturation of pro-B cell to the pre-B cell. So that is the correct sequence of events, that it begins with 3, then 1, 4, 2, and then finally 5. So therefore the correct option is option B. And finally, we were asked about somatic hypermutation of the heavy and light chain variable region genes occurs in which of the following zones. So we have discussed the way in which each of these events are sequentially, they occur in very spatially separated regions. That is, the light zone and the dark zone respectively have certain functions that different substeps of B cell maturation will take place in different subregions. So based on that, somatic hypermutation event occurs within the dark zone, and therefore option D is the correct answer in this case. So with this, we come to an end for the questions that were part of the previous week's assignment, and we'll quickly begin the discussion of the complement system. So this is just to give a summary. I did not want to keep it very wordy, but I think it helps you in quickly grasping the concept. So I kept it like that. So what is the complement system? What do you understand by it? The system by itself is considered to be made up of a large number of distinct plasma proteins, which are able to interact with each other. And what they do is they opsonize pathogens and they induce a series of inflammatory responses that help to fight infection. Obviously, if it is part of the immune system, it is helping you fight off some sort of an pathogen or some sort of an infection. How is it doing so? It has a cascade which is operational. The meaning of a cascade is that there are multiple players. You have a pipeline within which there are multiple players. The first one interacts with the second, then third, and so on and so forth. Another very good example of a cascade is the caspase cascade, if you're familiar with it, or the apoptotic cascade. Basically, these are cell death pathways, which are like the pathways are kept in place. It's obviously not always activated because the cell's default condition, based on the type of environment it is in, is to survive. But whenever there's a trigger that survival for the cell is not very beneficial and the cell should instead die, then under such circumstances, these cascades are triggered, and then the cell will obviously be subjected to these cascades. So I just gave that as an example. When we talk about the complement system, however, as we already said, its job is to fight off an infection. How will it do so? It will help in the opsonization of pathogens. What is opsonization? If you're already not familiar with it, it is the process by which you are taking a pathogen. So this is a pathogen. On its surface, you are forming certain pores. What is that pore going to do? It is completely disrupting the barrier between the pathogens insides and the surrounding environment, and all of the components which are inside of that living organism are coming out. Now, naturally, what that is going to do is it is going to kill off the cell. The way that this is done is it's an intricate process which we'll come to, but if you just conceptually think about it, you or me as an organism, if we just suddenly get like 1,000 pores on our body, obviously everything comes out, we are going to die, we are not going to live. And that is a simple principle which is used for most of the disinfectants or cleaners that we know everywhere around us. Detergents, for example, every detergent that you use, what it basically is doing is, it's obviously not opsonization, but what it does is it forms pores on the surface of the living organism, and that as a result will die. Or even 70% ethanol, which is commonly used for sterilization, in any research lab, that does something similar. It also has other roles, but one of the major factors is that it forms pores on the surface, and then the bacterial pathogens will no longer be able to counter it, and they'll die. So yeah, how the complement works is that it has a role to play in both innate and adaptive immunity. We'll come to this. How does it do so? In innate immunity, the complement system, it recognizes pathogens based on the recognition of the PAMPs, or the pathogen-associated molecular patterns. You're already familiar with this term because we had discussed it at the very beginning of the course, that the innate immune system by itself has the habit of recognizing patterns on the invading pathogen, one of which is via these PAMPs or MAMPs, which are pathogen-associated molecular patterns, or I forgot. PAMPs and MAMPs, which are on the surface of these pathogens, which are able to be recognized by the PRRs or pattern recognition receptors, and they help in early detection and early removal of the most common pathogens around us. However, in adaptive immunity, the complement action follows antibody binding of antigens on the pathogen surface, and therefore it is a more late onset event that after the adaptive immunity has been triggered, following that, there would be removal of these pathogens. So, yeah. So with this in mind, we have to understand the fact that a number of complement proteins are actually proteases that exist as inactive precursors or zymogens, and they get activated by proteolytic cleavage. What does that mean? Zymogen is an inactive enzyme. Any enzyme, this is not specifically with reference to the complement enzymes, but any enzyme for that matter are zymogens, and their cleavage helps in their activation. They go from their inactive or the precursor pre-enzyme stage to the active enzyme stage, and then they're able to execute their activity. There are three pathways of complement activation. First is the classical one. You're familiar with this already. Second is the ambilectin one, and finally the alternative one. The classical one is triggered by antibody or direct binding of the complement component CO1Q onto the pathogen surface. The ambilectin pathway is triggered by mannan-binding lectin, or the MBL pathway, a normal serum constituent that binds some encapsulated bacteria. And finally, the alternative pathway is triggered directly on pathogen surfaces. The classical pathway out of these is obviously, as the name suggests, or the definition suggests, it's the antibody-antigen interaction dependent form of complement activation. So we'll just briefly go over the type of experiments or the very early discovery of the complement system that was by Jules Baudet, and how Paul Ehrlich contributed to the same as well. So what did he observe? As early as in the 1890s, you can see that Vibrio cholerae, which is the pathogen responsible for causing cholera, when this was subject to the antiserum from sheep, and the sheep antiserum was specific to Vibrio cholerae, then that led to bacteriolysis, which means that these bacteria would die, and as a result would be lysed, and as a result would no longer be able to cause infection. Second is Vibrio cholerae. When it is mixed or when it is subjected to heat-treated sheep antiserum to Vibrio cholerae, then there is no bacteriolysis. However, when we have Vibrio cholerae, along with this heat-treated sheep antiserum, along with unimmunized sheep antiserum, then there is bacteriolysis. How is this possible? Very so what we understand from here is that there is some component in an within an unimmunized antiserum, which means that it is not the serum from an organism which has been exposed to that antigen, and therefore the antibody specific to it is present. No. Something else is present in the serum apart from the humor part of it. If you remember, the humoral form of immunity was actually dependent on the antigens that we were talking about. So apart from the antigen, from the antibodies, there is something else in the serum which is helping in the elimination of these bacterial pathogens. So what that is what they were trying to understand. So Baudet's simple test for detecting the lytic activity was by hemolysis, which is the lysis of antibody-coated RBCs. Now, what exactly this mystery system was, that term was coined by Paul Ehrlich, who described it as complement. There's a spelling error there, but yeah, complement, which is the activity of the blood serum that completes the action of the antibody. So complement as in the antibody or the humoral system which is in place for the immune response to be elicited is getting complemented by the help of this system. So what you need to understand now is that at the time of discovery, knowledge was obviously scientists were learning new things. And so at that time, the only way in which they understood the complement system was that it is helping or it is assisting the function of the antibody-mediated response. However, that is not necessarily the case. And instead, what we know now is that there are multiple pathways. But this is way back in the somewhere around 1890s or so. So at that time, based on the knowledge that they had, this terminology was coined. Now let us get into a little more detail. So these are the key steps that are present within the complement system. So after initial activation has been facilitated, what happens is that the various complement components are able to interact in a highly regulated or a highly controlled cascade, and they carry out a set of basic functions, which includes these following steps. What are those? So after there's an initial stimulus and there is activation of the complement system, there are a number of interactions or a number of participants within the complement cascade that interact with each other, and the execution is in these following forms. The first is via cell lysis. Cell lysis is in terms of the pathogen's cell lysis. That means that the cells are going to die. That is for cells, bacteria, or viruses. So you can see this is just again a schematic representation. This is not exactly how it happens, but you can see the complement activation is going to cause cell lysis. Cells are pores are being formed on the surface of the cell and they die. Second is opsonization, whereby you can see that bacteria is being engulfed by the phagocyte and it is getting cleaved off. Third is the activation of an inflammatory response. Just a second. Yeah. Lysis and opsonization are occurring. Okay, so I think I referred to opsonization differently in the past. I'll just opsonization is the coating. I was confused at that time. I apologize. What I was explaining at that time was lysis. Opsonization basically means that your pathogen is getting coated by antibodies and then it is getting recognized by the phagocyte, and then it is being taken up and is being completely removed from the system. Third is the activation of an inflammatory response, which is where we see that the complement receptor is triggering the degranulation of mast cells, which is releasing histamines. And then you can see that it is able to extravasate and move out of the blood vessel into the tissue, and it is able to execute its activity. And finally, there is clearance of the immune complex, which is majorly mediated by phagocytic cells. So that removes the immune complexes from circulation and is able to deposit them in the spleen and liver, which is completing the complement system's job. Next. So this is giving you an overview of the complement activation pathway. As we discussed, there are three different ways in which the pathway is getting triggered: the classical pathway, the MB-lectin pathway, and finally the alternative pathway. So the classical pathway is triggered when C1, we'll come to it. It is a macromolecule binding to the antigen antibody complexes, which is eliciting this interaction and the downstream cascade. However, the alternative pathway is getting initiated by spontaneously generated C3b. We'll come to that very soon. And that is the triggering antigen for that is different, and it is majorly based on the microbial cell wall antigens. The third lectin pathway, as the name suggests, is based on the detection of lectin by the immune system, and it is initiated by binding of serum pathogens MBL to the pathogen's surface, and all three of them are eventually converging to a step where C3 and C5 convertases are generated, and then they are helping in the formation of the MAC, or the membrane attack complex, by some sequential reactions. And if you look at the three pathways, the step at which they're converging is eventually coming to the hydrolysis of C3, which is the amplification step in all three parts, and that is generating C3b. So we'll come to that one by one. If you look at the classical pathway, obviously it starts off, because it's an antibody dependent interaction. It starts off with the binding of C1 to antigen antibody complex, which activates this molecule, and that helps in the conversion, and the formation helps in the conversion of C4 plus C2, resulting in the formation of C3 convert. And this underlined sort of denotion is what indicates the fact that it has already been cleaved and now is forming a complex of these two proteins. So this helps in the formation of the C3 convertase. The second MBL pathway is dependent upon mannose binding lectin, which binds to a foreign surface, and then it helps in the formation of the same complex. And on the third side, which is the alternative pathway side, we are able to see that it starts off with C3 and C3b. It is C3 to C3b formation is occurring at a very slow pace, and that is assisted by something called as factor B and factor D. We'll come to this later, which eventually helps in the formation of the complex, which is C3bBB. These are all the this is the compound which is formed as a result of all three types of cleavage, resulting in the formation of C3 convertase. Now, as we previously indicated, this is the major amplification step. And what is generated as a result of it, the final molecule generated as a result of it is C3b molecule, which you can consider to be as the the rate limiting step or the key driving step of this entire complement response pathways. Finally, after C3b has been formed, it is helping in the conversion or in the formation of C5 convertase, which, as the name suggests, will cleave C5 and will generate C5b. And from there, it joins or it integrates or it associates with C6, 7, 8, and 9 to finally form this sort of channel-like structure, which is able to embed itself onto the wall of the pathogen which it is targeting and clear it out of the system. What that will eventually do is all the cellular components will come out, the cell membrane integrity will be completely destroyed, and as a result, that pathogen will be eventually cleared from our system, and therefore, job of the complement system is now complete. Yeah, what we also see from the legend is that the C3b is also capable of diffusing away from the activating surface, and it can bind to other cell surfaces as well, and then its job will then become to serve as an opsonin. So if we go into the sequential events that occur as part of the classical pathway, then we'll come to them one by one. So the first thing you need to keep in mind is that the classical pathway is initiated by activation of the C1 complex. It is the it itself is also a complex, a macromolecular complex. What it consists of is C1q, two C1r molecules, and two C1s molecules. So this is what helps in the final formation. There was another, yeah. This will help you better understand. So this is the C1 macromolecule, which is, sort of a ternary structure that is generated as a result of all these associations. So this thick stalk-like structure is C1q, which is joined, whose globular heads actually are joined by C1r and C1s entities, which helps in the formation of the final structure. Each of these, if you look at it in more details, is made up of a collagen triple helix, and the globular head is able to bind to the CH2 domains of the antibody. Which antibody? The antigen-antibody interaction, which is triggering the complement pathway, is able to interact with this, and therefore it is linking, the complement pathway is getting linked to the adaptive form of immunity, or the humoral adaptive branch of immunity. So that is what makes this pathway dependent upon the antigen-antibody interactions, because if there is no association between the globular heads of all the C1q molecules and the antigen-antibody interactions which have already taken place, then the downstream cascade will not at all be triggered. So coming back here, what we see is this, the green representation is for antibodies, and along with that, you can see this reversed sort of a structure, which is representing the entire C1 macromolecule made up of C1q, R2, S2, and that is able to, the globular head of this molecule is able to interact with the constant part of the antibodies, or the CH2, specifically the CH2 part of the antibodies, and therefore it is able to elicit the classical pathway. Some additional information here is that a single molecule of IgM, IgM being the preliminary molecule or the preliminary antibody form or isoform, which is present and is very relevant with respect to the complement activation system, is capable of binding to, which when it is capable of binding to an RBC, it can activate the classical complement pathway and is able to lyse RBCs, while you can see some 1,000 molecules of IgG are able to do so. So basically what you see is that because IgM has the pentameric form, it is much more potent at executing the complement reaction compared to an IgG molecule, which has which will obviously suffer from a limitation when we talk about its efficiency in doing the same. And the reason for that is because the way in which you can ensure that two IgG molecules are close enough to each other that they are able to facilitate the formation of the C1QR2S2 complex is a lot more rare compared to an IgM pentamer, which is its stable form, and therefore it is more efficient at doing the same and is able to initiate C1q binding at a much quicker rate. So, yeah. So this actually shows you the different forms of the antibody. So this is the pentameric IgM in its planar form versus the staple form, which is the bound form. So circulating IgM molecules, they are actually present in this planar configuration, whereas you can see that the C1q binding sites are not exposed and therefore complement activation is not occurring. However, when pentameric IgM is bound to an antigen, which is when it has recognized its cognate antigen and it is binding upon the pathogen's cell surface and has found its cognate antigen, then it adopts this second sort of conformation known as the staple form, which you can see is a more pressed down sort of configuration. And there it has a lot more exposed sites where C1q can bind and the downstream classical pathway can be elicited. So yeah, now as you can see, C1q binds to its antigen bound antibody, and C1R is able to activate autocatalytically, activates the second C1R, and both of them are able to activate C1S. So this molecule is, this entire C1 macromolecule is now ready to elicit the pathway. If we go into a little more detail about the structure, if we go into a little more detail about the structure, just for reference, then none of this needs to be remembered, but just in case. So the formation of an antigen-antibody complex induces conformational change in the Fc portion of the IgM molecule, exposing a binding site for the C1 component. C1 in serum is a macromolecular complex, like we just discussed, and it is getting stabilized by calcium ion binding. It is made up of 18 polypeptide chains that are forming that collagen triple helix molecule that we saw. And the tip of that, or the globular head domain from those molecules, are able to bind to the CH2 domain of the antibody. Each C1R and C1S monomer again contains a catalytic domain and an interaction domain, and that facilitates its interaction with C1q to at least 2 Fc sites for a stable C1 antibody interaction to occur. Now, when pentameric IgM is bound to antigen, it assumes that staple configuration where, like we saw, which exposes 3 binding sites. However, in circulation, it is present in the planar configuration where those sites are not exposed. And like we said, the IgG molecule is much less efficient at doing the same job. So after this recognition has been facilitated, the next step is where C1S is cleaving C4 and C2. So here we saw that C1R2S2 were initially present. They are able to, C1R is able to act autocatalytically. It is activating the second C1R. Both of them are activating C1S. This is where step 1 is complete. Next step involves the action of C1S. One thing to keep in mind is that all of these enzymes, they are able to act as serine proteases, and they are basically when we say proteases, what they're doing is they're cleaving proteins. Like we said, all of the components of the complement cascade are actually proteins or peptides that are in circulation within the plasma, and they are normally present in the inactive state. Only when the initial trigger is in place or the pathway has been stimulated does its catalytic form come into play, and then it is able to keep on, each of them are able to keep on stimulating the next, and the cleavage products are getting released, and they're able to act on the next subsequent substrate. So in this case, C1S is next able to act upon C4 and C2. What it does is that cleaving C4 exposes the binding site for C2. C4 is able to bind the surface near C1. C2 also binds C4, forming the C3 convertase. The names might be confusing. What you effectively need to understand is that the final complex which is now getting formed is formed as a result of the association between C4 and C2 cleavage products. What are those cleavage products? You think of an entire protein which is getting cut off. The smaller part is A, the larger part is B. So the larger part of C4 and the smaller part of C2 are associating with each other, and the enzyme which is now getting formed is the C3 convertase. Again, the name C3 convertase means that it will now act upon C3, or its substrate is C3, and it will then form products, cleavage products from it, from which obviously, again, the smaller part is C3A, the larger part is C3B. I hope it is clear. It's a little wordy, but it's the simple action of an enzyme which acts on a substrate, generates two products, a bigger one and smaller one. The smaller one diffuses away in most cases. The larger one continues and forms part of the next enzyme in the cascade. So, now the C3 convertase will hydrolyze multiple C3 molecules. So that is why we kept saying that the key step or the key amplification step in the complement cascade is the step where C3 convertase is formed and C3 molecules are getting shut down, or they're getting cleaved. Not shut down, they're getting cleaved. Some of them can even converge, and they can act upon with the C3 convertase, and then they form the C5 convertase. So what it is doing is that it is positively regulating more and more activation of the complement system. So once C3 molecules have been cleaved and sufficient, or rather a large number of C3b molecules have been formed, they will then go and associate with the pre-existing C3 convertase to form the C5 convertase. Again, refer to the diagram if it is easier for you to understand. So this is the C3 convertase, which is acting upon its substrate C3 and is generating the larger cleaved fragment and the smaller cleaved fragment. Among these, the larger cleaved fragment, which is C3b, is then able to associate with this enzyme, which is C3 convertase, and is able to form a new enzyme, which is C5. Now, its entire name, if you go into the details of it, it has C4b, 2A, and C3b. All three of them are working together as a new unit now. What this will act upon now is that it will use C5 as the substrate and will generate cleavage products. We'll look at that in the next step. So, so here the C3 component, C3b component of the C5 convertase is able to bind C5 and therefore allows it to be cleaved. So C5 here is being represented as sort of a stick structure. The reason for that you'll understand in the next step, because it helps in the formation of the MAC. So C5 convertase is acting upon C5, generating C5a and b. C5b is the larger fragment, C5a the smaller one. In this case, what is relevant to us is again C5b. What it does next is that C5b is binding to C6, initiating the formation of the membrane attack complex. So C5b, C6, C7, C8 all together are forming this structure. So what you see at the center is C567 forming the central globular head-like structure, and all around it is the formation. What is represented here in pink is the assembly of the C9 part of the or the C9 protein or peptide, which is forming a sort of channel, which is going to form the entire pore, which can insert itself onto the cell membrane of the pathogen which it is targeting and forming the membrane attack complex or the MAC. This is then allowing the lysis and opsonization and the removal of the pathogen from the system completely for its eventual clearance. So this is a little more detail about the way in which the C3 convertase is working. So what you can see is the native form, or this is again a schematic representation only. This is the native form of C3 versus the activated form of C3, the way in which it works. So you can see it is being represented by two peptides, alpha and beta, which are bound to each other by disulfide linkages. Disulfide linkages, in case you're not familiar with it, are between two sulfur groups or sulfur side chains, which are usually present on the cysteine residues within these proteins, and they are able to form. They are usually necessary for formation of the 3D structure, and they help in increased stability of the protein. And therefore, their removal is a major way by which you can disrupt the structure of a protein. If you remember, we had talked about SDS-PH a few weeks back. So there we talked about the use of reducing agents. So reducing agents help in the removal of these sulfhydryl bonds, or these disulfide linkages, and would help in linearization of such a protein. So this is what you see here. Now this is the native form of C3. This is the native form of C3, and then can see C4b2A working here, which is a C3 convertase. And what it does is it cleaves off this very tiny fragment. I kept saying a small fragment, if you remember, right? So from the end here, like if you just second, yeah, if you think about it, it just gets cleaved from, sorry, oh, that was not correct. Just give me a second. Yeah, what I meant to say was here. So if this is the intact protein, there is a cleavage somewhere from here, and this part is what is called as C3A, and the rest of it is being retained as C3B, and now it is in the active form, which is then ready to act as an enzyme. And then what you see is the C3B can be bound also, and you can see that it is bound to a cell membrane. The membrane has been shown here. Uh, yeah, the membrane has been shown here, and then it can execute its activity. What happens here is that a labile internal thioester bond gets activated as C3b is formed, which allows this to bind to free hydroxyl or amino groups on the cell membrane. This is able to execute its downstream biological activities, which include C5 binding, so that it can then form the C5 which can then act as a C5 convertase, and then it can also bind to C3b receptors and phagocytic cells. So this is just a detail of the steps which are actually occurring, or the chemical reaction which is actually allowing these to act as enzymes, and what is activating the sudden catalytic activity in these proteins and allowing them to function as enzymes. So yeah, that was basically the complement pathway. So quickly summing it up, you have an antigen binding to an antibody, you have an antibody which is recognizing its antigen and binding there. Upon its binding, what is happening is that when that antibody, let's take IgM for example, which is a pentamer in that case. So that pentamer, when it is bound to its antigen and then it has found or is able to recruit the C1 macromolecule close by, then what it does is it triggers the downstream reactions. From C1, we can see that C1q, R, and S are sequentially activated. Then there is formation of the C3 convertase. The C3 convertase cleaves C3, forms C3A, C3B. C3B forms a complex with C5. Forms a complex to act as a C5 convertase, and then that cleaves C5, and then you have C6, 7, 8 forming the central pore, or the globular structure at the center of the pore, and then poly C9 assembling all around the side and helping in the formation of the membrane attack complex. So that was the basic idea that was followed, or that is followed as part of the classical pathway. Now, since this entire cascade is supporting the regular interactions between an antigen and antibody, therefore they are considered to be as complement systems, because they're complementing the action of an antigen antibody interaction, and they're helping in the immune response, which will help in clearance of the pathogen. Next, coming to the alternative pathway. So as the initiators of alternative pathway, they are a little bit different. Why these are particularly relevant, why you need to know about this, is because the alternative pathway, while we think about it as something, you might think about it as something which is sort of a backup. It's not the case. Both of these pathways, or rather all three of types of these pathways, are in place. They are operational. They exist within the system at any like they're present in the system at any point of time. It just depends what type of stimulus is coming in, which will dictate what type of a complement response will be the best or the most efficient at clearing out that infection as soon as possible. Now, when we talk about these antigens, which are being indicated here on the left, they are best removed or best recognized and best eliminated by the alternative pathway of complement activation. Let us understand what actually is encompassed as part of this. So the initiators of alternative pathway can be very broadly grouped into these two types. So we have pathogens and particles of microbial origin, and we have non-pathogens as well. So for microbial origin, we have many gram-negative bacteria, the lipopolysaccharides of gram-negative bacteria, gram-positive bacteria as well, teichoic acid from gram-positive cell walls. We have fungal and yeast cell walls, which is zymosan dependent. Some virus-infected cells are able to induce the alternative pathway of clearance. So again, you need to understand the fact that it's not always necessary that the alternative pathway of complement is what will eliminate a viral infection. No, of course you have NK cells, of course you have other pre-existing systems in place. This is just complementing the action of those and helping in quicker removal of the pathogen concerned. So Yeah, so basically that's why the immune system is working as a in a comprehensive manner, and it has multiple systems in place, so that in case one misfires or is not working efficiently, there will always be a backup in place. Again, despite that, we all do suffer from diseases, so it comes with its own set of limitations. But yeah, apart from that, some tumor cells can often also be, can also be recognized by the alternative branch or the complement system, and also for certain parasites, especially trypanosomes, which are a type of parasites. Trypanosoma, if you're familiar with it, is most a particular strain or a particular subtype of trypanosoma has been associated with sleeping sickness. So that's just an example. You might have come across it. It's very commonly given as an example in earlier years. Um, so these were the microbial origin pathogens and particles. For non-pathogens, we have human IgG, IgA, and IgE in complexes. We have rabbit and guinea pig IgG in complexes, cobra venom factor, heterologous erythrocytes from rabbit, mouse, or chickens, anionic polymers, and pure carbohydrates like agarose or inulin. So yeah, these if they're found to be circulating within our system, within our circulation, then they will initiate the complement pathway, and then they will be cleared off, and it will elicit an immune response basically, and they will then eventually be cleared off from the system. So how does this work? How does the alternative pathway work? You can see that again, the starting point is of course from C3. So you can see that from C3, its hydrolysis, the step where it is different is that its hydrolysis is occurring in a very spontaneous manner, and C3b fragment is able to attach to a foreign surface, to the foreign surface that we talked about. Again, if you have forgotten it, this is the reason why C3b is able to attach to a cell membrane or attach to an unfamiliar or a foreign membrane. That is because after it has been cleaved by the C3 convertase, it is resulting in the exposure or the activation of an internal thioester bond, which we saw just here, this thioester bond, which is allowing the C3b to now bind to the amino groups or the hydroxyl groups present on the surface of the pathogen under concern. When it does so, C3b attaches to the foreign surface, and after this, factor B is able to bind C3a, exposes the site acted upon by factor D, and this generates a C3 convertase. What is that C3 convertase? It has factor B, factor D, and the cleaved fragment from C3. So the resultant fragment, which is formed as a result, is C3b-Vb, and this serves as a C3 convertase. What this is doing is it is amplifying the amount of C3 convertase which is generated. So the initial event was a chance event, which means that hydrolysis occurred spontaneously. But once it has occurred, when you have a cascade which is purposely and consciously synthesizing more and more C3 convertase, it will amplify this signal. Now this C3 convertase can act on more and more C3 molecules, cut it, generate more and more signal, and then trigger the alternative form of the complement system, or the alternative pathway of the complement activation. So that's what you see here, that factor B and factor D are binding with it, and this is generating the C3 convertase, which along with properdin gets or helps in the stabilization of this convertase. Why is the stabilization necessary? Because it will help in triggering more and more C3b formation, and then that can go into a loop, and more C3 convertase will be synthesized, and then a large part of it will proceed forward for the removal of this or for the activation of the alternative pathway. So from the C3 convertase, obviously C3 comes in, it binds, it generates C3b, B, 3b. So what it effectively does, you can very calmly, you'll be able to understand what is happening there, that convertase is generating C3b. This C3b, this cleaved fragment, if you follow the cursor here, this C3 fragment is able to bind to the existing C3 convertase. This was C3b from this cleave fragment. This is where we started from. This associated with the factor B and factor D fragments, which is now forming the active C3 convertase. To this, we are again seeing an association of the larger fragment of C3b, or a larger fragment of C3, which is C3b. That is being denoted in this manner, and this is the final C3 convertase. It can be very easily be confusing. Please don't get confused. Remember that the converging step is C3 convertase. What you need to understand is the way in which you are arriving at the step where C3 convertase is synthesized is very different in the classical and the alternative pathway. That's just all you need to keep in mind. And for your own reference, what you should also, if you write this more, if you just make a simple flowchart, it will help you understand and retain this a lot better. So what you just need to do at that point is that you need to figure out the differences between those two pathways. What are the participants like? Obviously, factor B and factor D are not there in the classical pathway, or antigen antibody interaction is not there in the alternative pathway. So these subtle differences are what, not even subtle, these differences are what you need to keep in mind. And then what is the composition of the final C3 convertase, that you must remember. That can be a very relevant question. And also what you need to keep in mind is here that stabilization is taken care of by an external entity, that is property, which is also not the case for the classical pathway. So these differences are actually what will, help you in answering questions when you come to it. Finally, you can see that the C3 convertase and, the C5 convertase, I think it has been mislabeled. This is the C5 convertase. This C5 convertase helps in the cleavage of C5 into C5b plus C5a. And this C5b is what then participates in formation of the MAC, or the membrane attack complex. And otherwise, it can bind to the antigenic surface, it can permeabilize it, lyse it, remove it, and then clear it from the system. This is the entire pathway. So I hope you were able to understand it, but there's not a lot to explain here, very honestly. It's a series of steps which are occurring. There are components in place. They're just interacting with each other, cleaving each other, generating two types of fragments, and then that just does the job. So yeah, that's what we see there. Now, the last part that will be, not the last part, the second last part we'll be addressing today is that if all of this is in place, how do we keep it in check? I'll come to it. So what you understood from here is that you can think of the complement system like just a pair of random scissors, which can, cut and degrade anything and everything which is there in the system, and it will just clear any pathogen. The fact of the matter is, no. If that was the case, then maintaining the integrity and the specificity of the complement system would become an immense challenge. Therefore, its regulation or its control is extremely important, and there are several steps at which this is being maintained. So it's the more aggressive a pathway seems, the more important it is to ensure that its aggression is well monitored and well controlled and well regulated. That is what this regulation or regulatory check of the complement system basically does. So let's come to the legend first. So because many elements of the complement system are capable of attacking host cells as well as foreign cells. If you remember, we just saw that some of the antigens for the alternative pathway, they are human host cells themselves. Some of the antibodies, like I'll just go over to that slide. Yeah. So these non-pathogenic components, they are also very much considered as an initiator of the complement cascade. But if this was done in a non-specific manner, it would, of course, be very harmful. Therefore, that's where regulation becomes important. So elaborate regulatory mechanisms have evolved to restrict this complement activity to very, very designated, very specific targets. A general mechanism of regulation in all complement pathways is the inclusion of very highly labile components that undergo spontaneous inactivation if they are not stabilized by a reaction with other components. Additionally, there are a series of regulatory proteins that can also inactivate various complement components. Example being the glycoprotein C1 inhibitor. C1-INH is how it is normally denoted. It forms a complex with C1R2S2, causing it to dissociate from C1Q, and therefore it prevents further inactivation of C4 or C2. Let me just come here. Yeah, so this is the C1 inhibitor in black. What you see, it is just maintaining this protein in this in the C1R2S2 in that state, and is not allowing it to get cleaved. So that is what you saw here. This is the first regulatory step which is occurring. We'll come to this in detail, don't worry about it. But basically that is what it does. It forms a complex with this protein and dissociates from C1Q. So C1q is independently present, but C1R2S2 are not able to associate with it, and therefore the downstream pathway is not getting triggered. So the other proteins that regulate the complement system are as following. So you have the C1 inhibitor, obviously, which is a serine protease inhibitor. It causes this complex to dissociate from C1q. The second is C4b-BP, which is C4b binding protein. It's also a soluble protein. It affects both the classical and the lectin, MB-lectin, mannose binding lectin pathways. It blocks the formation of the C3 convertase, and by binding C4b, the cofactor for cleavage of C4b by factor 1. Then you have factor H, which is soluble and is part of the alternative pathway, blocks C3 for convertase formation. Then you have MCP, and you have complement receptor type 1, which are membrane-bound proteins, can act on all three pathways, and it blocks the formation of C3 convertase as well. Decay accelerating factor is DAE, membrane bound, accelerates the dissociation of C4b2A and C3b-BB, which are the C3 convertases. So if you look at it overall, what you will see is that the main step which you are targeting in most cases is that same C3 convertase formation or C3 convertase activity. And the reason why this is actually important is because, like we said from the very beginning, the C3 step is the most crucial, most rate limiting, and the fate determining step of the complement system. So that's what we say here. Apart from that, we have decay accelerating factor, factor 1, S protein, HRF, which is homologous restriction factor, or the membrane inhibitor of reactive lysis, or anaphylatoxin inactivator. I'm not going into the action of all of them, but yeah, you can read up on it. These are all the proteins that regulate the complement system. Now, if we quickly look at the steps at which this is done. So regulation of the complement system is occurring broadly, if you think about it, in two stages. One, before assembly of convertase activity, and two, after the assembly of the convertase. We'll go over them one by one. So before the assembly of convertase activity, how is the complement system being regulated or is being kept in check? First step is the binding of the C1 inhibitor, which binds to C1R2S2, causing its dissociation from C1q. Second, the association of C4b and C2a is blocked by binding C4b binding protein, complement receptor type 1, or membrane cofactor protein, which is MCP. So here you can see the association between C4b and C2a. This is C4b, C2a, which are together forming the C3 convertase. You are disrupting that. So you're not allowing C3 convertase to form. Next, inhibitor-bound C4b gets cleaved by factor 1. So what is it doing? That C4b, once bound by this inhibitor, is further getting cleaved. Such cleavage is resulting in C4c, C4d, obviously, because it has now been cut further. Once this has been done, you are completely terminating this entire step, and C4b can no longer associate with this complex and form C3 convertase. So you're stopping the complement reaction or complement activation cascade just at that step. In alternative pathway, CR1, MCP, or factor H, which we were just talking about, they prevent the binding of C3b and factor B. So this is C3b, this is factor B. Together, they are forming the C3 convertase. You are not allowing these two to bind to each other. C3 convertase is no longer formed. Alternatively, if it is formed, then the inhibitor-bound C3b will get cleaved to form C3F and IC3b. Again, factor 1 can act upon it, form C3c and C3dg, which means you keep on fragmenting the C3 protein further and further, so that C3 convertase can no longer be formed, and therefore that complement activation cascade will no longer be able to elicit its will no longer be active. Coming to the second chunk of the regulation, or the second section of the regulation of the complement system. Just a second, I'm getting yeah. So if we talk about the second section, which is for assembly of the convertase. So C3 convertases can either get dissociated by these proteins, C4b-BP, CR1, factor H, and decay accelerating factor. So that's what you see here. They are dissociating the two, and whatever is getting dissociated is getting cleaved. So C3 convertase assembly is getting disrupted. Third section is the regulation at assembly of the MAC complex or the MAC, which is the membrane attack complex. So this is C5b, 6 and 7. When S protein binds to it, it prevents its insertion into the membrane, and as a result, it cannot attack or cannot execute its action on any nearby cells. Alternatively, if you have HRF, which is the homologous regulation restriction factor, and membrane inhibitor of reactive lysis, which is MIRL or CD59, they are able to bind to C81, preventing the assembly of poly C9 and preventing the formation of the MAC. So what are the different steps we saw? First, at the level of the C1 inhibitor. Second, we saw multiple ways by which C3 convertase activity is getting inhibited. Downstream of C3 convertase, C3b is getting degraded, so that again, that step is completely stopped. Alternatively, we saw that assembly of C3 convertase is getting disrupted by certain inhibitors. Then we saw that after that activation has occurred and then the membrane attack complex formation needs to be carried out, at that step, the insertion of the MAC or the membrane attack complex into the cell membrane is getting stopped. And then finally, formation of the MAC can also be interrupted by these proteins. So those were the regulatory components of the complement system. Finally, if we talk about the biological effects of the complement system, obviously we have talked about opsonization and lysis being the effect, but what are the like if we have to segregate and then explain what exactly it is doing, that can be summarized from this table. So obviously the first effect is that you have cell lysis, and the complement product which mediates the same is the MAC. Second, you see the inflammatory response which gets triggered, and you can see all of the different proteins which participate in the same. The first being that there is degranulation of mast cells and basophils, which is mediated by C3a, C4a, C5a, which are anaphylatoxins. Second is the degranulation of eosinophils by C3a, C5a. Extravasation and leukocyte chemotaxis towards the inflammatory site. Platelet aggregation. Inhibition of monocyte macrophage migration and induction of their spreading. Release of neutrophils from bone marrow, release of hydrolytic enzymes from neutrophils, increased expression of complement receptors on neutrophils, and so on. Finally, we also have opsonization of particulate antigens, which is increasing their phagocytosis, viral neutralization, and solubilization and clearance of immune complexes finally. So all of these are the different types of biological effects, which is being mediated by each of these different subtypes of complement products, which is finally executing its activity. Yeah, so this is an example of the type. This is actually a SEM or a scanning electron micrograph. So SEM is I think I've introduced it to you before. Just a second. It's a form of electron microscopy which allows you to see the surface details of any living organism. So what you see are these E. coli or Escherichia coli, but these are bacteria. You can see these are intact cells with like perfect membranes. And in B and C, you see cells which have been targeted or killed by the complement mediated lysis. And you can see these membrane blebbing. So membrane blebbing is a major form by which cell lysis occurs. And that is eventually going to lead to cell death. So with this, we come to an end for today's presentation associated discussions. I will quickly go towards some just previous year sample questions for your preparation or for your understanding. If it is, I'm hopeful it is helpful because they are very similar to the assignment questions that you usually have. So that should, I assume, be quite helpful in that regard. So, okay, I can maybe go through this. If you remember at the very beginning of today's session, I had talked about the different types of interactions between cytokines. Now, I have not directly discussed this yet because you will be learning about the types of interactions between cytokines very soon. But yeah, basically, let's just read the question for our understanding. Two cytokines, IL-4 and IL-5, together act in the B cell class switching process. We have learnt about that already, how class switching occurs and what is the role of these interleukins or these cytokines. What is this phenomenon known as where both of them are together working for a certain process? A, redundancy, B, pleiotropy, C, synergy, or D, antagonism. Okay, I'll not go into details about this. You'll be learning what each of these means. But yeah, you might be already familiar with certain terms. Uh, Antagonism is where they oppose each other. You might be familiar with it. Pleiotropy is something I want to introduce with a diagram. But obviously, when two types of cytokines or two types of molecules for that matter are together acting in a certain process, they are helping each other out. If they are helping each other out, then such a process eventually is said to be synergized by the effect of both these molecules. So compound A, the effect which can be seen as a result of the action of compound A, result which can be seen by compound B versus the result which is seen when both A and B are present. If it is the additive effect of both of them, then that is said to be additive in nature. If you see that together, they are giving a much more heightened response, that by itself what A could have done, by itself what B could have done, versus when both A and B are together, they're leading to a much higher effect. Then such a condition is or such a situation is said to be synergy, that these two interleukins or these two cytokines are synergizing with each other. So that would be option C. If it is not something you're currently able to understand, don't worry. We will be discussing this when we come to cytokines. But yeah, you might be familiar with the term. So I just thought we could, take up this question. Now coming to question 2, that complements are the proteins that are involved in the clearance of antigens and bacteria. So initiation of which of the following pathways involves antibodies? A, alternative, B, classical, C, lectin binding pathway, or D, all of the above. So considering the extensive discussions we just had, you would obviously understand that it depends upon on the classical pathway depends upon the presence of antibodies, not the alternative or the lectin binding pathway. Therefore, the correct option is option B. Its initial stimulus comes from the antigen antibody interaction. So that is option B. Showing now. In question 3, you can see the major functions of complement proteins involve which of the following. So if you remember that initial schematic we had seen, maybe I can go back here. So yeah, these are the types of functions of the complement system. So we had lysis, opsonization, inflammatory response getting triggered, and finally the clearance of the immune complexes as all of the different actions that are executed by or the functions that are executed by the complement system. So all of these are present. So if you look at the options, option A, B, and C, all of them are correct. Therefore, option D would be the correct answer in this case. Coming to question 4, which of the following is not a C3 convertase? So we saw the different types of C. This is why I told you that you need to just, for your own understanding, tabulate and write down the differences between the different substeps of each of these pathways. And I think that should be good enough for you to remember. Coming back to the question, we've been asked which of the following is not a C3 convertase: C4b2a, C3b Now, see, for example, it can get a little confusing because it's a lot of numbers and just A and B being written about. If that is the case, if you are not able to remember the correct option, if you're not able to remember the correct names of the enzymes in each steps, if you just go through the options, you'll very, very clearly be able to see that there is one option which is definitely not correct, which is option C. C1q R2S2 is the first step. If you remember the entire C1 molecule, macromolecule that we talked about, which is made up of these R2 and S2 subunits as well. And we had seen how the C1 inhibitor interrupts this assembly and completely can shut down the downstream process. So therefore, option C is definitely not the correct answer. So that's why I'm saying you do not necessarily always need to remember the subtle details, but if you have the conceptual clarity that C1q is the first enzyme, C1q is what will trigger all the downstream steps, you'll be able to answer questions like this. And here it's just 1 point, but yeah, you get questions like this. And, if you eventually want to go for research, if you go for the All India competitive exams, or even, interviews in person, or for that matter, the degree for this for this course as well. These questions hold a lot more weightage, and skipping these questions where you can attempt answers with some basic concepts, you just don't want to miss out on answering such questions. So just be careful, be mindful when you study, that you know the idea that how is this system working. Some things are rote learning. If you're not able to remember them, it's okay. You can skip such questions. But yeah, these are very easy. Please do not skip answering questions like this for fear of negative marking, because you'll be missing out unnecessarily on some good marks. Yeah, coming to question 5. Which of the following complement cleavage products is not a component of the MAC or the membrane attack complex? So we have C5b, C6, C9, and C3b. Again, see, this is a very easy question. Even if you don't remember what the MAC is made up of, you know that it's definitely not C3b, because C5b has to come in, and from C5b the assembly starts. And C3, C3b are the entire process of production of C3b is the main amplification step. It is where, like I said multiple times, it's the rate limiting or the driving step, and that is where um, that is where all three pathways converge and they help in production of more and more C3b. So that is not a component directly of the MAC, but it is an intermediate which you can consider to be the most important intermediate in the complement system cascade. Uh, Coming to question 6. Which of the following components are part of the lectin pathway? So there is A, mannose binding lectin, B, phycolin, C, membrane associated serine protease, or all of the above. So yeah, I did not get into details about this, but if you go to, just give me a second. Yeah. I think it was in the overview, most probably. Yeah. So if you come here, my cursor is not there, but yeah, if you look at this topmost part of the schematic, you'll see that the lectin pathway of course has the mannose binding lectin, or the MBL, which helps in triggering or stimulating the lectin dependent pathway. It is cooperating or coordinating with the MBL associated proteases, which are MASP1 and 2, which generate a complex very similar to the C1QR2S2 complex. So what is formed in the classical pathway, analogous to that, something is formed in the lectin pathway as well. So both the mannose binding lectin and the associated MASP, or the MBL associated proteases, both of them are operating in this process together. So that is definitely there. And phycolin, while I have directly not mentioned it here, phycolin is also a form of lectin. It is um, it is an antigen which can stimulate the lectin pathway of complement activation, and therefore all three are correct. Therefore, correct option is option D, all of the above. Uh, Coming to question 7, chemokine receptors belong to which family of cytokine receptors? Yeah, this you don't need to get into right now. We have not discussed this yet. This is because of the previous year syllabus, its structure was different. So you don't necessarily need to know it, but just in case you're familiar with it already, if you've studied GPCRs at any point of time, you would know that these CXCR family of receptors, which are the chemokine receptors, they are all GPCRs, or G protein coupled receptors, which means that they are surface receptor molecules. So at the cell surface, there are these receptor molecules present. They are G protein coupled, which means that there's a receptor, and very close to it, there is another small protein known as a G protein. What is the relevance of this? You have your receptor, your ligand is binding on top of the receptor. The downstream G protein, which is present, is then executing the remaining activity. How that works, we can get into that when we discuss cytokines if you're not familiar with it. But yeah, G protein receptor signaling, or GPCR mediated signaling, is very interesting, very common, very abundant throughout the system. And yeah, you can look it up if you have already studied it at some point of time. Else we'll discuss this in the next few weeks. We only have a few weeks left for the course, so we'll be discussing that then. And that is the correct answer. That's why I went into so much detail about it. Which of the following complement cleavage products can enhance inflammation and help in neutrophil extravasation? So you have C3A, C5A, C3B, C4B, C3F, C3C, or all of the above. So yes, I'll just go to that. Yeah. So this is actually where you'll get your answer. Again, like I said, some things are rote learning. It's just you can conceptually remember that which step has the cleavage products of which step are involved in which reaction, but it's difficult. That's why I did not unnecessarily stretch too much on it. If you can remember, please do. Please go ahead and look into it. It can be a curious question that in every subsequent step, we are seeing that every time a protein gets cleaved, you are having a smaller fragment and a larger fragment. You can see that the larger fragment continues in the complement cascade, but then what happens to the smaller fragment? It is actually doing all of these things. If you see here, most of the complement products you will see here are A. So C3A, C5A, the C4A for that matter, all of them are going and doing the, they're executing the parallel reactions which are helping the complement response. So one of them is the interaction with neutrophils. So you can see here that they release neutrophils from the bone marrow, and they release hydrolytic enzymes from the neutrophils, and then they're able to execute their activity. So that's why I gave this chart so that you can look and refer to it for your better understanding. So yeah. So in this case, the correct answer would be C3A and C5A, which is option A. Which factor cleaves the membrane-associated complement cleavage products C4b and C3b, leading to their inactivation? So that was factor 1. I think we had seen this here. Again, I'll refer to slides because easier to remember. Yeah. So this is factor 1 or factor I, however you wish to pronounce it. So again, I don't have my... Yeah, so this is factor 1. So that actually operates in all 3 pathways, and you'll have this as it acts as a serine protease, or it functions as a serine protease, which cleaves C4b or C3b, and it executes its activity. We also, I think, had it here. Yeah, in this diagrammatic representation, also you can see that factor 1 is involved here, and it is able to do its job. So this is also a very direct factual question. So you just need to remember the answer to it. There's no shortcut to that. So yeah, that is question 9. And for the last question, which is about the regulator of the complement that can bind to C5b67 and inhibit innocent bystander lysis, is decay accelerating factor, homologous restriction factor, S protein, or the membrane cofactor protein. So here it is option C, which is S protein. I don't think we have taken it up directly. Let's just go back to the slides. This is talking about regulators, right? Yeah. I did not directly mention it, but yes, it is there in the slide. So S protein is binding to soluble C5b67, and it is preventing its insertion into the cell membrane. So what is mentioned here in the question is about innocent bystander lysis. It is inhibiting that. So that's not being mentioned here. You don't need to worry about it right now because you don't directly have this addressed as part of the syllabus right now. But for your own knowledge, you can keep it for your own reference. So yeah, with this, I am coming to the end of today's class. If there's any question, you can please go ahead and ask. But basically, we summed up the entire complement system. We looked at all of the different components, all of the different regulators, initiators of the pathways. We looked at the regulators of those pathways. We looked at what is the biological effect of those of the cleavage products of those pathways, and also looked at previous year's questions. We addressed last week's assignment, and yeah, basically gave a comprehensive overview of week 9. This week, we'll be looking at week 10 and obviously in the subsequent weeks 11 and 12. And then finally, we'll have an extra session. I think you will also have an interaction session if you've not had it already with the course instructors, a live interaction session with them for your better understanding. Please try to attend that because interacting with the course instructor is always a good idea. It will help you clear out more doubts. They're obviously they're experts in this field. We are also learning in this regard. So please don't miss that session. And if there are any doubts, please go ahead and ask. Drop your questions in the discussion forum. Somebody will definitely address them. And yeah, all the best. I will be uploading this video and the slides as well in the Google Drive link, and you can access that at any point of time. And yeah, please let me know if there are any doubts, and then we can end the session for today. Okay, if not, then I'll end the session for today. Thank you so much for joining. Um, Hope you have a great night, and I hope these sessions are and the video lectures and everything is very useful for you. Study hard. Your exams are pretty soon, and all the best. Thank you so much for joining. Good night.